This video is going to be a continuation of the history of our understanding of DNA video, but this video is going to specifically focus on the Hershey-Chase experiment. So after Oswald Avery discovered that DNA was responsible for the transformation of bacteria, that was a good bit of evidence that DNA is the genetic material and not any of the other major molecules in cells. However, a lot of people still were under the impression that it could be proteins. Proteins are highly variable. They basically are the expression of our genetic code. Why couldn't they be our entire genetic code? And so Alfred Hershey and Martha Chase specifically focused on comparing whether it's protein or DNA that is our genetic material. They used viruses called bacteriophage viruses. I think they look pretty awesome, like lunar landers over here. Bacteriophage basically means bacteria eating. So they used these viruses, and you should know about viruses. Viruses are basically a protein coding. Okay. So they have a protein coat and some sort of nucleic acid inside. In this case, it was DNA. And on the outside, we have protein. So Hershey and Chase grew viruses in cultures with either radioactive phosphorus or radioactive sulfur. And you should remember from our biochemistry unit that of NCHOPs, DNA, and all nucleic acids have N-CHOP, and protein has N-CHOS. Okay? So this phosphorus and sulfur is a big deal. If I grow viruses in cultures with radioactive phosphorus, it'll only end up in the DNA. If I grow them in radioactive sulfur, it'll only end up in the protein, which is great. And so we can radioactively label either the protein or the DNA and follow where it goes. And so the substance that ultimately was injected into the cell was DNA. And we'll look at this. Here, in this first experiment, you can see that they have a glowing green protein coat because the proteins here were radioactively labeled with that radioactive sulfur. Okay? The viruses are allowed to infect some bacteria, and at the end, they look at the cells and say, okay, is the radioactively labeled item inside the cell or in the environment outside the cell? And you can see here that the phages, those protein coats, stay outside of the cell. Meanwhile, looking at DNA here, we have now radioactive DNA using radioactive phosphorus. You can see that the glowing piece is on the inside here where the DNA is. Again, phages are allowed to infect bacteria. And yet again, we look at the end and say, okay, where is our radioactively labeled substance now? It's inside the cell. And so this proved to everybody that DNA is actually our genetic material, not proteins.